So tonight is a webinar being brought to you guys by the Subcortical Surgery Group. It's going to focus on immunotherapy and newly diagnosed glioblastoma, results from a phase 1b trial, and then talking about the new phase 2b trial with Dr. Andrews. Just real briefly, the Subcortical Surgery Group, or the SSG, is really a users group of surgeons and practitioners dedicated to optimizing minimally invasive corridor surgery to the subcortical space through really clinical and scientific collaborative research, technology applications, all for the betterment of our patients and really leading these changes in safe and effective minimally invasive treatments. And so we feel that in addition to this corridor-based surgery and some of the other things technically we're doing, a lot of the things that you'll hear tonight from Dr. Andrews in terms of immunotherapy and optimizing kind of the surgical and postoperative management for patients with glioblastoma, this speaks to that as well. It's really trying to do the best for our patients and minimizing any morbidity. So if you're interested, you can register, you could scan this QR code. We have a great yearly meeting that's obviously been on hold for the last year or so. It's oftentimes in Boulder, and it's just a really nice day-and-a-half meeting, a lot of interesting people, great talks, and it's a good opportunity. So if you're interested, that's the website, that's the Twitter handle, and we'd love you guys to join us. Just upcoming webinar plug here. This is with Mike Shagru, absolute star, and such an intellect, but this is going to be informing neurosurgical disconnections using connectomics. This is a multi-part webinar that has a hands-on component using some of the software that Dr. Shagru has developed, harnessing the connectome, and really is going to, I think, revolutionize the way we think about brain tumor surgery and ablative brain tumor surgery, and it's a really good one. So if you have the time, I'd certainly encourage you to participate in that. So again, today, going to be focusing on immunotherapy and newly diagnosed glioblastoma, and we have the honor of having Dr. Andrews here, exceptionally experienced brain tumor surgeon and researcher, did his training in New York, doing residency at Cornell, and did fellowship at Sloan Kettering. He's been really researching the immunologic phenomena behind glioblastoma for decades. He currently resides at Thomas Jefferson. He had started the division of neuro-oncology and is its chief, and then spun off to start a company, Imvax, really harnessing a lot of the research that he has been instrumental in developing. And he's going to tell us about the results of their phase 1 trial, and then the exciting phase 2 trial that's going to be upcoming, and hopefully is going to have a massive impact in this disease. So I'll stop sharing so Dr. Andrews can share here.
Sure.
Just make a quick plug. We have the Q&A function active. So if you have questions, please put them in the Q&A. When Dr. Andrews is done, I think we'll have some time to be able to address some of those questions and engage the group. So please don't hesitate.
Thank you, Brad. So I'll begin. So again, it's really an honor to be involved in the subcortical surgery group. I am the chief medical officer at Imvax and hold financial interests in the company, but I'm still a full-time professor at Jefferson. So I don't want to move too quickly, and I'm going to frame all of this from the eyes of neurosurgeons. I think most of us on this webinar are neurosurgeons. So we're all familiar with glioblastoma as a really a horrible orphan disease with unmet need. And for that very reason, this, the CTGA was really the first tumors that they looked at was GBM because it was so intransigent. And from that became a lot, really, it begat a lot of really interesting new information. And Cameron Brennan at Sloan Kettering really capitalized on that in at least a molecular understanding of what's going on. However, the other side of that is, instead of from the bottom up looking at the gene or the transcriptome, we have to look at it from a different standpoint, which is the tumor itself and how we can treat patients. So this is more of a top-down talk, and particularly we're going to talk about what I've been doing for 20 years. So generically, there are a number of different paradigms, and you're probably familiar with all of them. 2019 was not a good year for glioblastoma, so checkpoint inhibitors with actually BMS. Every checkmate trial was actually negative. And then if you drop down to antibody drug conjugates, the ABT414, which is the AbbVie trial for newly diagnosed GBM patients with EGFR overexpression, that was negative. And actually, what is in terms of oncolytic viral therapy, Togagen 511, which looked promising for injections of a virus, genetically engineered virus that was transforming a prodrug into an active drug, which is an old active cancer drug, 5-fluorouracil. When that went to a prospective randomized trial, they did not meet their primary endpoint. So as we gathered information from our phase 1b trial, we were looking, and this was actually a KOL meeting I had in November of 2019, which ironically included, actually he was the chief medical officer at Moderna. Little did he know what was ahead, but kudos to Tal Zaks for what he achieved. But he said, look, based on 2019, your next step has got to be from a phase 1b trial. You've got to go to a prospective randomized placebo-controlled double-blinded trial. And I took that to heart immediately, and that's where we're going. Monoclonal antibodies. I'm not going to dwell on this slide, but it's just very clear that there's a lot of interest and a lot of different approaches to this disease. I would say in very general terms, targeting one antigen, two antigens, maybe even up to three antigens is not necessarily going to give us success, because this particular tumor, as is the case with just about all solid tumors, is they're very complex. And it turns out that when one looks at the body's response to these tumors, whether it's glioblastoma or hepatocellular carcinoma or pancreatic cancer, the infiltrating lymphocytes, they've actually isolated and sequenced the T cell receptor to reverse engineer what they thought might be common antigens, and it's just not the case. 98% of non-synonymous mutations in these tumors are unique to that patient in that tumor. So this was published in 2013 on the heels of the revolution in treatment of cancer with checkpoint inhibitors. And this is really a good general sort of scheme of what an ideal immunotherapy would do. So tumors release cancer antigens at the bottom of this clock. Antigen presenting cells will then take up these antigens. They'll then traffic to lymph nodes where they then interact with naive T cells. The T cells then traffic back through the circulation with diapedesis, in this case in the brain, to the site of a glioma, where these now activated T cell populations will attack the tumor, and cytolytic interaction with the tumor will then release more antigens, and then you get this self-perpetuating cycle. That's the ideal. So I'll kind of revisit this this scheme after we go through some of the preclinical data and what we've learned. So when we look at glioblastoma, we have a number of constraints we have to deal with. The blood-brain barrier. I don't know why this is automatically doing this. This is an immunologically cold tumor, so it's only 2 to 3 mutations per megabase, which is really unlike non-small cell lung cancer in smoking patients, or gosh, I'm sorry, this is just doing this, or melanoma, where UV light is creating mutations. GBMs create an immunosuppressive environment systemically, and we have documented elevation of M2 macrophages, which are normal in our healing process, but M2 macrophages, it turns out, are detrimental as they're drawn into the tumor microenvironment as an immunosuppressive cell population. And then finally, the final challenge we have is that we still have to deal with standard of care, which radiation alone causes immunosuppression, particularly lymphopenia. Now, the slide is going forward. I hope it's not on an automatic. So here is just an example of a lot of different solid tumors. And to the very far right are the tumors I described, which are typically responsive to checkpoint inhibitors because they're hypermutated, either because of the carcinogens that smokers inhale into the lung, or to UV light, which fuels lots of mutations in melanomas. So you can see where glioblastoma is. So in this case, we're looking at non-synonymous mutations, which represents mutations that create structural changes in the translated protein, which could then be immunogenic. And you can see that this was work done by Castle, it's Dana-Farber. And really, the vast majority of solid tumors, 98% of the proteins that are mutated are unique to that individual tumor in that patient. And that's relevant to what I'm going to discuss. So just one footnote is that solid tumors widely express the targeted protein that we start this process with, which is the insulin-like growth factor type 1 receptor. So it's a surface receptor that is really central to our biology as mammals. It regulates growth, cell division, control of metabolic activity, and is anti-apoptotic. So going back to the M2 macrophages, they overexpress the insulin-like growth factor type 1 receptor, so they have a longer half-life than M1 macrophages. So you can see here that IGFR1 overexpression is rampant in most tumors, solid tumors. And thus, actually, this could be actually a platform treatment for tumors beyond glioblastoma. So what is this process? This is a process in which, to the right in this cartoon, we're all familiar as neurosurgeons with the treatment of glioblastoma patients. So newly diagnosed patients, standard of care begins with craniotomy. We remove tumor, and intraoperatively we send tissue to pathology to get a definitive diagnosis. Typically, though, the remainder of tissue is disposable. Patients are usually discharged by the 4th postoperative day and then proceed with adjuvant treatment up to 42 days later. So what we've been able to do is, with equipoise, is interpose a process in which we take what was otherwise disposable tissue. If we get a frozen section diagnosis of glioblastoma, we actually then have prepped out the abdomen. And again, as neurosurgeons, most of us still open the abdomen. Some of us actually will engage our general surgeons to do a laparoscopic placement of a peritoneal catheter, but I think we were all trained with opening the abdomen down to the down through the posterior sheath to implant a silastic catheter. In this case, all we have to do is go down to the rectus sheath and open it and create pockets between the sheath and the muscle, which is the cartoon that you're seeing to the left. So the tumor cells, after we get a diagnosis, are actually all collected in a process, and this is maybe the plug for subcortical surgery. We usually try and either go with an interhemispheric approach or always splitting a sulcus and minimizing really a transcortical resection. I have often used the Myriad, the NICO Myriad device, because it's an FDA-approved means of collecting, morcellating and collecting tissue that's viable, unlike either Sonopet or CUSA, which is also quite effective, but ultrasonication will actually cavitate cells and potentially cells that would have much less of a yield. So we need a cell, we need a volume of cells, and we need those cells to be viable. So those cells are then collected during surgery. We proceed with craniotomy, and after the frozen section, as we're closing, During hemostasis, we open the abdomen on both sides and create pockets between the sheath and the muscle. We do a temporary closure, the patients are recovered, and the following day, the patients are implanted with this process. So you see in the interposing tumor cell processing phase, as in the first 24 hours after surgery, the cells go to a BL2 facility where they're plated. Eventually, they're encapsulated in small biodiffusion chambers, which actually is something the FDA insisted on. They didn't want us redistributing cancer from one place to another, even though glioblastoma doesn't do well outside the central nervous system. And these were chambers that have a subcellular exclusion limit. We spike the chamber with the API, which is an antisense molecule to the insulin growth factor type 1 receptor, which in and itself is actually very important in this process. Those chambers are then sealed, irradiated, because again, the FDA wanted irradiation to be absolutely sure that we were not going to redistribute cancer. And they're implanted the following day, generally in our institution in an intensive care unit with actually conscious sedation, usually Versed and fentanyl and local anesthesia. It's very well tolerated. It takes about 35 minutes. The chambers remain in the patient for 48 hours, and then we come back and explant them. And the patient is without complication that we all have from unforeseen complications from surgery, which is generally small. They're usually out on schedule by day 4, well within the DRG, which is now almost 6 days. And then, of course, adjuvant therapy thereafter. So IGV is the product which now is under development through this company we've developed. So it's a whole tumor immunotherapy. So it's taking the full array of antigens from that tumor in that patient and creating this product. We've talked about the process. We'll go into a little bit about what if we unpack this combination product, which we call IGV-001. That's, we think, is what is actually giving us the clinical signal that we're now translating into a phase 2 trial. So this is kind of a cartoon summary of what I just discussed. So it's not antigen agnostic. So instead of, for instance, targeting the epidermal growth factor variant 3, or in the rare case of a BRAF V600E mutation, or actually survivin, or any other overexpressed antigen which may provide a potentially an actionable handle for treatment. This is antigen agnostic, which means that the full array of antigens coming out of that chamber are what the immune system is going to see. And that's what we believe is creating the likelihood of a anti-tumor pro-inflammatory response. So it's a multivalent antigen approach. The API, which is an 18-mer oligonucleotide, which I'll review in a second, is actually the drug or the chemical that we actually spike the chamber with. And I'll go through that in a moment. So it's a multidimensional strategy that in the course of this process, with the antigens that are released and the excess oligonucleotide that we spike the chamber with that diffuses out of the chamber, we're actually then activating dendritic cells, and it turns out probably B cells that probably have seen tumor antigens, and thus we have mature B cells that recognize antigens, as well as the antisense, which is now targeting M2 macrophages that are systemically overrepresented in glioblastoma patients and are probably in this local environment. So in sum, we've created a wound, we've implanted a foreign body, so that's immediately drawing the interest of the innate immune system, out of which is diffusing this oligonucleotide, which I'm going to, as I describe in a moment, itself immunostimulatory. So it's an adjuvant in this immediate environment that the innate immune system is responding to. So then the dendritic and or B cells are going to then traffic to the, in this case, probably the inguinal lymph nodes, where they're then going to be marshaling platoons of T cells, each of which are responding to different epitopes. And then going back to the cancer immunity cycle, they're then going to traffic to the tumor site. So the challenges that we face, all of us collectively, is the blood-brain barrier. So just based on what I was just saying, the disruption of the blood-brain barrier after a resection of a tumor, based on TBI literature, should remain open for up to an hour, which provides a highway for an initial adaptive immune response. I mentioned the challenge of standard of care, which we all know causes immunosuppression, particularly radiation and lymphopenia. Temozolomide as well can cause pancytopenia. So based on meta-analyses from the RTOG before it was rolled up to the NRG, there was no difference between 2, 4, and 6 weeks. So we were able to push treatment to 6 weeks, adjuvant treatment, to allow an immune response to occur. And as I mentioned, this is a multi-antigen approach. As such, it's actually, we've seen no off-target effects that one might see with a checkpoint inhibitor or even CAR-Ts, where you can unleash a cytokine storm. We've seen nothing like that. And this is simply recapitulating what I've already described. So this is the oligonucleotide. So within the oligonucleotide, which is an 18-mer, this is the antisense molecule, there's a CpG motif. So this dinucleotide is what is otherwise referred to as a pathogen-associated molecular pattern that the innate immune system is already pre-programmed to recognize. So as antigen-presenting cells come into this area, they're immediately seeing an environment in which they're reading a CpG motif, and they're interpreting this, because it's unmethylated, as a massive bacterial infection. So the antigen-presenting cells are then taking up antigens, which are assumed to be of bacterial origin, but in fact, it's the full array of antigens we described earlier. The TLR9 agonist is then causing the antigen-presenting cells to mature, whether the dendritic cells or B cells, to then traffic out of the battlefield onto the draining lymph nodes to then interact with naive T cells. So there are many actually additional immunostimulatory features of this molecule. So beyond the CpG motif, there are palindromic motifs. Now, what's interesting is you'll see a palindromic motif in the sense or the antisense sequence. So the sense sequence is what is typically going on biologically in our cells. So that's important because as I go through some preclinical data, if it were just these immunostimulatory motifs, then sense should actually perform the same way as antisense. So this is a paper we published in 2015, and man, it's 6 o'clock and people are wondering when they're going to be able to get dinner. So I don't want to go through too much of this. All you need to take away from this is what we learned is that in an animal model, because we're all kind of stuck, or at least we all start as neurosurgeons, and quite frankly, many people in cancer research start with preclinical models. So this is the GL261 model in C57 black 6 mice, which actually is a reasonably good model. Every model has its drawbacks, and one must always take preclinical data with sort of a grain of salt, because it doesn't necessarily translate into human immunity. However, in this case, what we have found here translated into what we then developed in actually the clinical trial. And all you need to know here is that GL261 is naturally immunogenic. It was actually a carcinogen that created a GL261 cell line from a mouse brain. So it's immunogenic, perhaps for no other reason, because it's a brain tumor, which the peripheral immune system of the mouse doesn't typically see. So you see that if you treat patients with either Nobel or Nobel sense, or I mean mice, you see no difference in survival. So PBS mice, of course, develop no tumor until you challenge them, and then they respond. So there was no difference between sense and anti-sense. However, if you then admix the GL261 cells in a flank with antisense, then you see this striking improvement in survival of these mice and lack of tumor progression, which is not seen with sense. So this is just simply showing, number one, that it is very important to actually admix tumor cells with this molecule in vivo to get an effect. And then finally, to the right, here, rather than admixing the antisense molecule with the tumor cells, we put the tumor cells on in one flank and the antisense in the other, and you see that you lose the effect. So it's very important that the antisense and the tumor cells remain together to create an immune response. So going back to the cancer immunity cycle, do then these cells cycle, do they then traffic? Well, they do, because if you take ipsilateral lymph nodes and look at markers of immune response and activation of the adaptive system, the draining lymph node, you see a really striking response. In the non-draining lymph node, you see nothing. And you see nothing with Nobel sense or the sense sequence. And the same is true. So think of this as B cells, and this is dendritic cells. So we're seeing two antigen-presenting cell populations responding to this treatment. And then finally, do we actually see an interferon gamma response in this model? And of course we do. So all you need to know here is that the surgical control shows far fewer interferon gamma spots in an ELISpot than the implantation of this combination product. So this is getting really complicated, but this is now all the data that I've just reviewed and more that shows you that we have at multiple levels the capability of pushing the immune system from a tumorigenic environment to an immunogenic anti-tumor environment. So here's the phase 1b data that we just published in Clinical Cancer Research. It came out on April 1st. So I designed 4 randomized cohorts in an otherwise single-arm trial. And you'll see that we have an even accrual of, you can't do a typical dose escalation as you would with a drug in a 3+3 design. So I had to design an exposure to what I thought was a process of antigen release. So it was 10 chambers all the way up to 20 chambers at different time points, 24 or 48 hours. And you'll see here that the highest dose accrued much more because we had exploratory objectives that looked at immune responses. And we found early on that there were far more robust pro-inflammatory responses in this highest dose cohort. So we continue to follow these patients, and by patient 23, those patients were doing better clinically and radiographically, so we stopped the randomization. So these are the results, and this is based on a data lock of May 1st of last year, which is holding. So if we look at the intention to treat population and survival, we're seeing nothing actually for the entire intention to treat population. Now that's important because this was a phase 1b trial in which actually I was advancing the hypothesis that if there's an systemic immune response, it doesn't matter how much disease burden there is in the brain, we should see a response. So I included bihemispheric disease, multicentric disease. I didn't stipulate age. I didn't stipulate a Karnofsky score lower. I mean, actually I did stipulate a Karnofsky score of 60. and I did not stipulate extent of resection. So when you actually then peeled away 11 patients who would not have met criteria for the original Stroop trial, this is what you see. So you're now beginning to see a separation if, in fact, this trial enrolled only patients that would have met the Stroop trial and all subsequent control arms based on the Stroop trial. And then when you go to the highest dose cohort, then you're looking at a significant difference. Now, we all understand that this is a phase 1b trial, and slicing and dicing data with such a small population is really treacherous. And I'm the first to actually, I'm not a shill. I mean, I'm just, we were just seeing results that many people were saying this is unusual. So for reference only, we're looking at control arms. The control arms here represent not historic populations, like a matching population at Jefferson, for example. There's a footnote at the bottom. There was a publication out of the UK in 2012 in which there's a program in which you can actually digitize published log rank survival curves. And you can take those data and extract those endpoints. You can't get patient-specific data, but you can actually get endpoints. And that's what we did here, and we got over 1,000 patients from 3 different trials for comparison. When looking at progression-free survival, it was even more impressive. So we had actually, for the entire intention to treat population, for Stroop-eligible patients, and finally for the high-dose patients, we were seeing significant separation. So when we actually look at PFS, again, simply rolling up all of these arms, sorry, this was actually death from tumor progression. That's why that's so significant. So even for intention to treat populations, we had a higher rate of death than, for instance, the Stroop trial. And we think that's because the progression-free survival is much longer, and thus the morbidity and mortality from standard of care, and we saw this in this trial, was creating a higher incidence of morbidity and mortality. So it's the standard of care arm with longer progression-free survival that we think is that's reflected in this. So again, if we look at progression-free survival, all the way across the board, we're seeing a significant separation. And then finally, methylated patients, we're actually seeing a very striking difference. In this case, this is 319 patients in the Stroop Palindromic trial that was segmented to look only at methylated patients. So these are the radiographic responses. To the left, gross total resection, and to the right, subtotal resection. I was more interested in the subtotal resection, though having said that, we found really durable responses in the gross total resections. So this is all published in World Neurosurgery. So if gross total resection is followed, median time to progression is published at around 9 months. These patients have not yet reached that. And in the subtotal resection, which I found more interesting, residual disease is what I was interested in looking at, particularly even in the most challenging cases of multicentric or multifocal disease. Now, we didn't see that. In fact, I will not include multicentric or bihemispheric disease in the phase 2 trial for that reason. There's a biology to a disease burden which exceeds really our understanding of actually what's going on. So it has to be unifocal disease, or it can be actually multicentric disease, or I'm sorry, multifocal disease. We will still include them in this upcoming trial. So here you see examples of subtotal resection out of the plane, for instance, on the right in row A. There are other areas of enhancement. This is this young man's dominant hemisphere, so we only went after this lesion. But what we found is that there was a complete regression through all axial planes, all of this to essentially non-measurable disease, and that was true in other patients. So again, this was another patient, and this was another publication of World Neurosurgery. subtotal resection was essentially a median time to progression of 6 months, and here it's, for these patients, it's almost 33 months. So these are the exploratory objectives I was referring to. This is actually figure 5 in the clinical cancer research article. So we were looking at pro-inflammatory cytokine responses after this treatment. And these are just 6 examples of patients that had perfectly synchronized pro-inflammatory responses for interferon gamma, IL-12p70, and IL-2. And using nanostring, we looked at gene pathways and found actually an elevation in responses for both for antigen processing signatures, including chemokines, cytokines, macrophage function during a pro-inflammatory response in the TNF superfamily, and interestingly, out to 90 days. So what we're planning actually is different from what I'm showing you. So this is something that we designed last year. And Brad, this is probably something you have seen. So this was going to be a phase 2b randomized trial with a 2-to-1 accrual. We were leveraging progression-free survival based on the data I've showed you. And secondary endpoints would have been overall survival. We have since obtained the capability of going much further in terms of what we hope to be a pivotal trial. So we're going to be in front of the FDA with a Type C meeting, and this is going to be a much larger trial. So this is going to be a pivotal phase 2/3 trial. It's going to be 300 patients. Progression-free survival will be our first interim analysis. And it's actually going to be powered for a hazard ratio of 0.5. Originally, we had a 90% confidence level. We're actually going to go to a 95% confidence level, which gives us an opportunity to look at not only a first interim analysis, but a second, if we don't meet significance actually at the first interim analysis. And then of course, we'll ultimately look at overall survival. So this first patient first visit in will be probably September now of this year. And in the interim period, we're going through this process with the FDA, as well as qualifying cell processing centers. One that we're building 4 clean rooms in a 17,000 square foot facility at Imvax, and we have another CDMO in Boston, which is why we're limiting ourselves to a radius from those 2 areas. And regrettably, we were originally involving the West Coast and the Midwest, but we realized that it was just going to be impossible to get cGMP production at every site at an academic institution. So we've taken control of that. So it's now 6:41, so we're actually 4 minutes ahead of schedule. These are my colleagues. I'd like to certainly recognize Kevin Judy, who was actually my collegial friend, even when he was at Penn. We would always second each other's second opinions. He left Penn and came to us over a decade ago, and he's actually been very involved in this trial. And Craig Hooper, whose translational team, when it was all really an investigator-initiated trial, was really quite phenomenal. So I'll stop there. We're on schedule. I know that's a lot.
Yeah, that was phenomenal. I think for somebody who's not far cry from a tumor immunologist, I think your initial just description of the challenges that we face with glioblastoma and the basics of tumor immunology as it applies to this trial were as succinct and I think digestible as I've heard in a long time. So certainly appreciate that, and I'm sure the rest of the participants feel the same way. Got some questions coming in. The first is from Dr. Slavin. It asks if there's any data to compare your results from the phase 1b, from the phase 1 trial, I assume, with some of the results from the checkpoint inhibitor studies.
We haven't. I guess because they were negative results. They were it was. I mean, of course, the checkmate trials were huge. We could easily have done that because they were negative trials. I think we went with the ones we went with, which actually we took the two bevacizumab trials because we were actually processing all of these data before the readouts for checkmate. I think if we included checkmate, I don't think overall survival, progression-free survival were terribly different. I mean, they didn't meet their primary objectives. So.
Yeah, no, absolutely. And I'm always curious, overall survival versus PFS. I know with some of the original, with the bevacizumab trial, for instance, it looks so impressive when you're looking at PFS, but when you look at overall survival, you tend not to see that. Seems like that hasn't been quite the same issue with the immunotherapy. We're seeing these really maybe remarkable radiographic responses that don't necessarily correspond to survival, but I know that always comes up. And so how'd you guys ultimately settle on PFS?
So it's a great point, Brad, because it was for that very reason that bevacizumab sort of sank PFS as a primary endpoint for glioblastoma, because the responses that both the Gilbert and the Chenot trials had, really there was striking improvement in progression-free survival. We now realize that was pseudo-response. And it was just really rearranging the biology of these tumors, which is why they didn't reach their primary endpoint with overall survival. We saw initially, and we anticipate not a direct correlation. I don't think we powered overall survival in this phase 2/3 study as a hazard ratio of 0.6 with a confidence level of 80%. And the reason it's less is because of standard of care. So we are still faced with a treatment which is based on the response of the patients, the immunologic response of these patients, which is then challenged by standard of care. Yeah. And thus, overall survival is mitigated not only by the morbidity of standard treatment and what it does to the immune system, but additionally, all of the subsequent treatments, which are open field for subsequent treatments, whether it's going to be tumor treatment fields or quite frankly Avastin or whatever else patients may go into as an experimental trial, which makes overall survival, it's the ultimate endpoint, but it's mitigated by so many other variables it's hard to interpret, and thus it's not a linear relationship with progression-free survival.
Yeah, no, that makes sense. So another question coming in from Leo Ampe. So it says, you mentioned the role of plasmacytoid DCs and B cells, and since B cell activating factor increases after Temodar, are you tracking these cells in the post chemotherapy time point? Is the question.
So this is only preclinical data. As we approach our phase 2/3 trial, we're loading our exploratory objectives with those very issues. So we're doing quantitative analysis of fluctuation in cytokines and serum cytokines. We're looking at different white cell populations after treatment. So in addition to doing next-gen sequencing. So we don't have really reliable biomarkers for response. So really we have to use surrogate markers to assess not only what we think is an immune response, but also what that immune response might correlate with in terms of how the patients are doing.
Yeah, no, that makes sense. Yeah, speaking of that, in the phase 1, did you guys notice any more robust, like pseudo progressions, kind of these inflammatory responses that are maybe sometimes seen in patients getting systemic immunotherapy for brain metastases, for instance, where you'll see a really kind of robust inflammatory change? Did you guys see any of that?
That's a great question. And amazingly, we didn't. And patient 3 in that right panel of subtotal resection was a prime example. I mean, his 6-month scan was scary. I mean, it was a consolidation of all of those left opercular areas into one large consolidated mass of enhancement, which by any standard was progression. But for the fact, and again, we take Irano, it's not something the FDA accepts, but it's certainly in this era of immunotherapy, something that we all look at. So as we look at our phase 2, we're using RANO criteria, which we're adapting. So in this particular case, that patient did not have any signs of progression. He was off steroids and actually training with his father for the Big Sur Marathon. So based on IRANO criteria, we felt this is not progression. And that was true for a number of other patients.
Yeah, that's so interesting. So again, throw it out to the participants here. You know, please, again, we don't, any questions you have, feel free to feed them through the Q&A and get Dr. Andrews' input on kind of what stands to be a really extraordinary trial. I think when you first look at this or you hear about it, it strikes you as odd. And I think to hear from you kind of the rationale behind all the separate little parts that make up this trial, it all makes sense. The abdominal portion, the implantation of the biochambers, the irradiation of the chambers. I mean, all these things I think come together in a novel paradigm to hopefully make an impact in this disease where as you pointed out in your initial slide, really not much has worked. This is a unique approach.
Thank you, Brad. It is a Kitty Hawk flight. We're getting air. But I think there are probably going to be many iterations after this, which will make this more elegant as we understand this better, this process.
Yeah, well, this is a great start. We certainly appreciate your time. And I think, but I know our shop's going to be hopefully participating in the study. I think it's an exciting one. I'm sure everybody's going to be looking forward to seeing this materialize and progress. And fingers crossed.
Fingers crossed.
Well, great. So if there's no other questions, we'll maybe give everybody back a few minutes. And again, want to thank everybody, on behalf of the subcortical surgery group, and obviously thank Dr. Andrews for his time and participation. Certainly, I think you have contact information for the subcortical group. Any follow-up questions, certainly feel free to reach out to us. And don't forget about the other upcoming webinars with Dr. Shagru on connectomics. Again, also stand to be of great interest.